using technology to bring you affordable medicines faster. How many of you have walked into a pharmacy to get a medicated cream and were told the price of a product and its cheaper alternative? Or better yet, for example, told that a 200 Rand cream would do the exact same thing as a 14 Rand cream. Suspicious, isn't it? I don't blame you, because we are used to regarding cheaper alternatives as knockoffs, but that is the beauty of generics. They are not knockoffs of the original products. In fact, they are the exact same. Getting a new drug onto the market is a complicated process. Even for generic companies, they invest a lot of time and money in order to get their products onto the shelves. At the heart of this journey is the ability to demonstrate that a generic drug has the same chemical properties and same effects on the patient as the original drug. We call this bioequivalence. When you think of drug testing, the first thing that comes to mind is clinical trials in human subjects. For example, we've seen this with the COVID-19 vaccine clinical trials. However, this is not the only route available. For certain drugs, such as topical creams, which do not enter the body but rather work locally on the skin, we can use the power of technology to help companies that manufacture generic drugs get their products onto the pharmacy shelves sooner, which will ultimately help you as a patient save money quicker. Since this is the goal of my project, I have the task of getting as close to a human clinical trial as possible using only laboratory equipment. With clinical trials, the human subjects would have the generic and the original cream applied to the skin, and for each subject, the release rates of the cream would be measured, the data gathered, and at the end, a comparison made between the generic and the original cream. This means I have two problems if I want to simulate this scenario. The first problem is I have to simulate the behavior of the cream on the skin, and the second problem is measuring the release rates of the cream into the skin. To simulate the human skin, I use a vertical diffusion cell. Sounds complicated, right? But simply put, it is made up of a membrane which mimics the skin and fluid beneath it that mimics the layer of the skin in which the drug will collect. In essence, each vertical diffusion cell is a human subject. I might have solved the first problem, but now I need to find a way to measure the release rates. To do this, I collect the sample of fluid, which in essence is much like collecting the samples from the human skin to assess how much drug is passed into it. By the end of the research, my aim is to demonstrate the bioequivalence of an original and generic drug using only technology, thus facilitating more affordable healthcare.